guys, today's video is about the microbial structure. The major groups of microorganisms, namely bacteria, archaea, fungi, algae, protozoa, and viruses, has its own different structures and functions. There is the two types of organism, the prokaryotes and eukaryotes. Let's head first to the prokaryotes. The organisms that belongs to prokaryotes are the bacteria and the archaea. First is the bacteria. Bacteria comes in 5 basic shapes: the spherical, cylindrical, coma shape, corkscrew, and spiral. The scientific names for these shapes are cocci, bacilli, vibrios, spirochetes, and spirilla. The shapes and configuration of bacteria are often reflected in their names. Archaea has similar structure as bacteria. The only difference is archaea has rectangular or irregular shape. Now, let's proceed to the inner structure of these organisms, which is the prokaryotes organisms. So, as I said earlier, that organisms has its own different structures and function. So, in this structure, you can see there is a capsule. Some species of bacteria have a third protective covering, a capsule made up of polysaccharides. Capsule play a number of roles, but the most important are to keep the bacterium from drying out and to protect it from phagocytosis by larger microorganisms. The capsule is a major virulence factor in the major disease-causing bacteria, such as Escherichia coli and Streptococcus pneumoniae. Non-encapsulated mutants of these organisms are virulent. They don't cause disease. The cell envelope. The cell envelope is made up of 2 to 3 layers: the interior cytoplasmic membrane, the cell wall, and in some species of bacteria, an outer capsule. So these 3 layers here are called the cell envelope. Next is the cell wall, the green layer. Each bacterium is enclosed by a rigid cell wall composed of peptidoglycan, a protein sugar molecule. The wall gives the cell its shape and surrounds the cytoplasmic membrane, protecting it from the environment. It is also helped to anchor appendages like the pili and flagella, which originated in the cytoplasm membrane and protrude through the wall to the outside. The strength of the wall is responsible for keeping the cell from bursting when there are large differences in osmotic pressure between the cytoplasm and the environment. Next is the cytoplasm. The cytoplasm or protoplasm of bacterial cells is where the function for cell growth, metabolism, and replication are carried out. It is a gel-like matrix composed of water enzyme, nutrients, waste, and gases, and contains cell structures such as ribosomes, a chromosome, and plasmids. The cell envelope encases the cytoplasm and all its components. Unlike the eukaryotic cell, bacteria do not have a membrane-enclosed nucleus. The chromosome, a single, continuous strand of DNA, is localized but not contained in a region of the cell called the nucleoid. All the other cellular components are scattered through the cytoplasm. One of those components, plasmids, are small, intrachromosomal genetic structure carried by many strains of bacteria. Like the chromosome, plasmids are made of a circular piece of DNA. Unlike the chromosome, they are not involved in reproduction. Only the chromosome has the genetic instruction for initiating and carrying out cell division or binary fission. The primary means of reproduction in bacteria, plasmids replicate independently of the chromosome and, while not essential for survival, appear to give bacteria a selective advantage. Plasmids are passed on to other bacteria through two means. For most plasmid types, copies in the cytoplasm are passed on to daughter cell during binary fission. Other types of plasmids, however, form a tube-like structure at the surface called a pilus that passes copies of the plasmid to other bacteria during conjugation, a process by which bacteria exchange genetic information. Plasmids have been shown to be instrumental in the transmission of special properties, such as antibiotic drug resistance, resistance to heavy metals, and virulence factor necessary for infection of animal or plant host. The ability to insert specific gene into plasmids have made them extremely useful tools in the field of molecular biology and genetics, especially in the area of genetic engineering. Another is a cytoplasmic membrane, a layer of phospholipids and proteins called the cytoplasmic membrane encloses the interior of the bacterium, regulating the flow of materials in and out of the cell. This is the structural trait bacteria share with all other living cells, a barrier that allows them to selectively interact with their environment. Membranes are highly organized and asymmetric, having two sides, each side with a different surface and different function. Membranes are also dynamic, constantly adapting to different conditions. Next is the flagella. Flagella are hair-like structure that provides a means of locomotion for those bacteria that have them. They can be found at either or both ends of a bacterium or all over its surface. The flagella beat in a propeller-like motion to help the bacterium move toward nutrients, away from toxic chemicals, or, in the case of the photosynthetic cyanobacteria, toward the light. Next is the nucleoid. The nucleoid is a region of cytoplasm where the chromosomal DNA is located. It is not a membrane-bound nucleus, but simply an area of the cytoplasm where the strands of the DNA are found. Most bacteria have a single circular chromosome that is responsible for replication, although a few species do have two or more. Smaller circular auxiliary DNA strands called plasmids are also found in the cytoplasm. Next is the pili. Many species of bacteria have pili, small hair-like projection emerging from the outside cell surface. These outgrowths assist the bacteria in attaching to other cells and surfaces, such as teeth, intestines, and rocks. Without pili, many disease-causing bacteria lose their ability to infect because they're unable to attach to host tissue. Specialized pili are used for conjugation, during which two bacteria exchange fragments of plasmid's DNA. Then, the ribosomes. Ribosomes are microscopic factories found in all cells, including bacteria. They translate the genetic code from the molecular language of nucleic acid to that of amino acids, the building blocks of proteins. Proteins are the molecules that perform all the function of cell and living organism. Bacterial ribosomes are similar to those of eukaryotes, but are smaller and have a slightly different composition, and molecular structure. Bacterial ribosomes are never bound to other organelles as they sometimes are in eukaryotes, but are free-standing structure distributed through the cytoplasm. There are sufficient differences between bacterial ribosome and eukaryotic ribosome that some antibiotics will inhibit the functioning of bacterial ribosome, but not eukaryotes, thus killing bacteria but not the eukaryotic organism they are infecting. Next is the structure of the eukaryotes organisms. The organisms that belong to eukaryotes are the algae, protozoa, and fungi. This is the structure of the protozoa. So as you can see, in this cell, the cell is enclosed in a membrane called the plasma membrane. Like all membranous structure in the eukaryotic cell, the plasma membrane is composed of mostly lipid and some protein molecules. The plasma membrane is a barrier between the cell cytoplasm and the outside liquid environment. Cilia and flagella arising from the cell are also sheltered in the cell membrane. This is in contrast to bacterial flagella, which are not surrounded by a membrane. The cell also has internal membranes, which are not as thick as the plasma membrane. Among these are the endoplasmic reticulum, whose membranes separate compartments of the cell, thereby allowing different conditions to be maintained in various parts. The separation of deleterious reactive substances. Enzymes are arranged on the surface of the endoplasmic reticulum. One such enzyme system catalyzes the activity of the ribosome during protein synthesis. The Golgi apparatus is a cluster of flattened vesicles or cisternae associated with the endoplasmic reticulum. The vesicles are involved in membrane maturation and the formation of the storage of the product of cell synthesis. As in the formation of scales on the surface coat of some flagellates, example, the scales are formed within the Golgi and are transported by the vesicles to the plasma membrane, where they are incorporated on the surface of the cell. The Golgi apparatus is poorly evident in most ciliates and absent for some amoebae. Next is the structure of algae. So algae have a cell wall, and this cell wall is composed of cellulose and other substances that gives the algae form. And inside the cell walls of eukaryotic algae are three main types of organelles: the nucleus, the chloroplast, and the mitochondria. There are several other organelles present in eukaryotic algal cell, including the endoplasmic reticulum, Golgi apparatus, lysosome, and vacuoles. The reproductive cells present in algae are unique because they move with the assistance of thread-like appendages called flagella. There are many different types of flagella which vary in size, shape, and arrangement. Now let's proceed to fungi. In almost all fungi, the hyphae that make up the thallus have cell walls. A hypha is a multi-branched tubular cell filled with cytoplasm. The tube itself may be either continuous throughout or divided into compartments or cells by a cross wall called septa. The last one is the virus. Virus is not belong to eukaryotes nor in prokaryotes. Viruses are much smaller than bacteria and consist of single or double-stranded nucleic acid surrounded by a protein shell called a capsid. Some viruses also have an outer envelope composed of lipids and proteins. They vary in shape. So viruses has capsid RNA envelope protein spikes, and membrane protein. Learning the structure of microbes helps in understanding how a microbe functions. That's all for today. Thank you for watching.